Everybody, welcome to the second to last Warnell Seminar. So a lot of y'all on our if you're a student on the schedule, we had like a debrief day. And there was a visiting PhD student from Spain that wanted to present to us. I figured, do we really need to debrief? Why not have another great seminar? So we're gonna have another seminar on November 14th, maybe, I think, that Thursday. Sounds about right. And then there's not gonna be a final debrief. So just make sure you're doing the same number of posts, and then there's kind of like a final reflective post based on a couple things you learned, different speaker styles that you like, and then also like suggestions to make the seminar series better. So do that. Make sure you mark that you're here. I'll pass this around, and then I'm gonna let Katarina introduce our speaker.
All right, everyone, it's well, welcome to the semi-last seminar. At this point, I was already ready to say, last one, that only beers and we're done. But no, sorry, I'll have to wait more. Okay, so it's my honor to have here today Dr. Jeff Garnas, who's an associate professor of forest ecosystem health at the University of New Hampshire in Durham, where he has been since 2016. We're just saying today, we started at the same time. We started there, I started here, so we're same age, academic age. All right, so Jeff is a forest ecologist whose work focuses primarily on biotic disturbances in forest ecosystem, principally those caused by non-native and or range expanding insect and pathogens that attack canopy trees. He has a master's from the University of Maine and a PhD from Dartmouth College, both in ecology and evolution. And prior to working in New Hampshire, he was a faculty at the University of Pretoria in South Africa, where he spent nearly 7 years deeply engaged with countries, diverse ecosystem, and cultures. Jeff work retains global focus, but is now more deeply rooted in the forests of the Northeast US, ranging from the northern hardwood forests of Hubbard Brook to the pine barrens of Long Island. And with this, Jeff, I'll leave it to you. Thank you so much for coming.
Well, thanks, Katarina. Thanks for inviting me. It's been a real pleasure meeting folks today. So, your beautiful campus, which is immensely larger than the campus that I come from. Okay, so thanks all for being here as well. So, yeah, I'm going to talk about biotic threats to forest sustainability in an interconnected and changing world. So I like to start with a picture. This is a picture I took from the top of a mountain called Blue Joe Mountain. It's not much of a mountain. It's about 500 meters above sea level at the top there. But it's nice because it looks out over the seacoast of New Hampshire, which is where I live. And it looks like quite a forested state. It turns out that New Hampshire is the second most forested state in the nation. It's about 81% forest, only surpassed by Maine. But this is all secondary forest. It's very much here a human-dominated landscape. It's a little bit hard to see, but there are roads running all through that, in little towns and villages and homes. But and much of the land ownership is private in this part of the world. But this is all secondary forest. This was consequence of land abandonment about 150 years ago. So this would have been all farms and pastures. And so it was about 20% forested 150 years ago, and now it's 81% forested. So all these forests are approximately the same age. There is still harvesting, but let's put about a 150-year age on these forests. So, but a lot going on. So I know this is the Warnell School of Forestry, so you all know about forests and the key services they provide, but of course forests regulate things like all important things like climate, hydrology, the air and water quality, and biodiversity. 60 to 80% of our terrestrial species are found in forest ecosystems. Forests also provide abundant resources. So of course there's timber and other wood products. You guys are well familiar with that. We've been losing mills at sort of a scary rate up in New Hampshire, but we still have an active harvest, partly for timber products, but also much for firewood. So firewood, pulp and paper, some non-forest products like mushrooms, forest nuts, syrup, medicinal and foraged plants. It also is really important as a habitat for wildlife. So a lot of our forestry effort goes into conserving forests as a way of serving our native bird and mammal species, and hunting is obviously very big here. And then there's recreation and cultural and aesthetic value. And forests can be a sustainable resource for us, which is one of the reasons they're so valuable to us. But then the question is, what is a sustainable forest? Maybe lots of people thought about this, and it's one of those topics that's sort of easy to think about, but hard to really define exactly. But when we have a forest, forests are inherently, as all ecosystems are, subject to some different forms of disturbance. So we have abiotic disturbance like fire and wind and drought, flooding, ice damage, freeze-thaw cycles, et cetera, that can push the forest away from its sort of equilibrium state, if you believe in equilibria. We have biotic disturbance, so native insect outbreaks, of course, pathogen pressure, herbivory, seed predation, all things that are natural in our native forests. And then we have anthropogenic disturbance, so harvesting, salvage operations, transient land use change. All these things tend to push the forest more or less to some earlier successional state. There's a little bit of controversy in that statement if you read the literature, because we don't necessarily proceed through the successional dynamics the way it was originally conceived, but generally speaking, that's the case. And then the forest has a natural propensity to return to its original state, a closed canopy forest, roughly dominated by the species that are the most shade tolerant in our part of the world, which would be beech, maple, hemlock, depending where you are. Yeah, and so this is just a succession dynamic graph, and so we don't imagine that these forests move back all the way to the pioneer stage, but cycle somewhere in this zone as a function of how much disturbance there is, how frequent it is, and the severity of that disturbance. And the disturbance is also an important part of these ecosystems because it promotes biodiversity. For example, one of the reasons that we need to manage our forests in New Hampshire actively is because as people remember the forest, it was a relatively young forest, and that was amenable to a lot of early successional bird species, moose. For example, in a closed canopy forest, moose and deer don't actually do that well. So if we want to maintain moose as a part of our biodiversity and as a hunting asset, we need to maintain early successional forests. But then there are, of course, perturbations to the forest that can lead to much more dramatic change. So, for example, acid rain was a really important part of the ecology of a lot of the northern hardwood forests in our part of the world, led to spruce decline and really reduced sugar maple growth and recruitment. That was shown quite nicely in a calcium addition that we did that was done at Harvard Brook, not by me, that recovered a lot of the sugar maple regeneration health in that experimental forest. Changes in fire regime, fire exclusion, especially in the eastern forest, and then in western forest fire is much more important. There is some evidence that fire was part of our ecosystems, especially as utilized by Native Americans. Certainly not as much of an issue as it is out west, where you can get set back and force the ecosystem to more of a cheatgrass dominated system. And then there are a whole suite of evolutionarily novel biotic threats, and mostly I'm speaking here about non-native insects and pathogens. So that's sort of where I've put the focus of my lab since I've been here, and even before that at the interface of the ecology, evolution, and management of novel biotic threats, forest ecosystems. And friend Yen Ashmore that you guys know well, beech bark disease, which I'll talk kind of extensively about. If you want to look at my lab, you can take a picture there, but you can also probably find me fairly easily. So many people probably know that we've been experiencing a steady accrual of insect pests and pathogens globally, really, but certainly in the US, and this is a paper from a few years ago now, but represents a more or less linear increase in the number of non-native forest insects, about 2.5 new forest insect establishments per year since the middle of the 18th century, and about half a pathogen a year. So one new pathogen every 2 years during that time period. So that's a lot. And of course, once they establish, they then spread. So we're now seeing a situation where a lot of these pests and pathogens are starting to co-occur in places where they didn't before. And we happen to be sitting in one of the most invaded areas of the US. New York is actually takes the #1 seat, but New Hampshire is in an area where there are lots and lots of forest invasive. And this is really a consequence of global connectivity. And of course, we have this set against the backdrop of climate change. So climates are changing. We live in a warming world. Everyone knows that, and to some degree that can be correlated with the invasion of non-native pests and pathogens, but also just it changes our systems, both native and exotic. So we're seeing dramatic change, right? We have the mountain pine beetle, native insect changing dramatically as a consequence of range shifts and elevational shifts as a function of climate change. We see climate change influencing our forests and other landscapes. Non-native, this is myrtle rust or guava rust, which is my representation of an exotic pathogen. There are many I could have used. And all of this is linked to global connectivity, primarily is driven by global trade. And all of these things are interacting in very important ways that are difficult to disentangle, of course, but we need to do that if we want to understand these systems. So in many ways, we sit at the interface of historical, active, and emerging threats. So here's a picture of chestnut blight, representation of a historical threat whose legacy we probably barely really understand, since chestnut has been more or less deleted from our forests, with some exceptions. And we have our newer invaders like the emerald ash borer, and I'll talk at some length about the beech leaf disease nematode. That's our newest forest threat, or one of the newest anyway. So as a forest health practitioner, I guess, I think a lot about some of the perturbations that can lead to more dramatic change. So for example, in some cases, we've seen the rapid loss and replacement of foundation or ecologically unique species. So chestnut blight is a good example. So here's a picture of chestnut in all its glory around 1920. And here's me standing next to what's putatively the largest chestnut known in New Hampshire. And you can tell the scale is quite a bit different now. And most of the chestnut is just little stems that emerge from the root sprouts that have survived. So there's still hope for chestnut, but that's a whole nother story and probably involves genetic modification, so we won't go there this particular seminar, but that's an example of a really dramatic change. And then we have things like hemlock woolly delegate that has killed many thousands, millions of hemlock trees, and it's just sort of rearing its head in New Hampshire. It's still limited a little bit by winter cold where I live, so it's not quite as dramatic, but in North Carolina and some parts of this, Appalachians, it's really been a devastating loss. And now the emerald ash borer, of course, is really changing a lot of ecosystems and ash-dominated ecosystems across the U.S. People see talks about this particular pest, I'm sure. So generally speaking, this loss of biodiversity can lead to a loss of functional redundancy and ultimately to reduce resiliency in our forests. So that's kind of the concern if we keep losing species. When do we start losing function, ecosystem function, and then when does resilience begin to decline? We expect fully that ecosystem services of various sorts are also lost as species of importance to ecologies and socioeconomics decline. And that is represented in this figure here from a paper in 2013 that I don't think we need to dissect, but different pests and different forest systems are going to have different subtle changes in the loss of ecosystem services that are incurred. For example, hemlock was really important, or is really important, as a regulator of stream temperature in the water that moves through and hydrology in systems that are hemlock dominated. And once we lose hemlock, stream temperatures tend to increase and trout suffer. Other cold water species tend to suffer, fish species and aquatic insects as well. But really the one that we most worry about is system state change. And there aren't that many examples of system state change in eastern forests. There are some, so basically what we're talking about here is this idea that there are either alternative stable states. So the equilibrium dynamic might be, this is just a conceptual graph, obviously, but you're up here somewhere, and if you keep pushing the system with some sort of stressor or driver, that if you push the system far enough, it can eventually switch into a different stable state, and it's very difficult to get it to switch back. So, and this can also be conceptualized as this kind of ball and cup diagram, or as a regime shift. So do we have examples of alternative stable states in forest ecosystems? There are a few. So for example, black ash seasonal wetlands in the presence of emerald ash borer and to switch to permanent alder swamps. Might be able to say the same thing about the red brand brooch beetle in certain systems down here. I'm less familiar with that in its native habitat, but you guys might know about that. Unmanaged pine barrens, we're seeing in addition to fire suppression, in addition to the northern range expansion of the southern pine beetle, has the potential to lead to oak scrublands. So loss of pitch pine as a canopy dominant, And then certainly in the southern Appalachians, where chestnut blight has already reduced biodiversity in the tree canopy, add the hemlock woolly delegate, and we see some examples of rhododendron thickets emerging that are sort of at least stable at a decadal time scale. So these kinds of changes we really worry about. So talk a bit more about that, but as an outline, I'm basically going to talk about the topic of forest sustainability and novel interactions in a changing world. And what I'd like to do is give you a little bit of an overview of some of the projects going on in my lab, kind of like almost a slide per project, just to sketch out the range of projects that we're working on. And then I'll talk a bit more in depth about beach bark disease in North America. That's the one I've spent the most time studying, and about beach leaf disease as an emerging threat. All right. So just briefly, there's my family. I decided to leave that picture in there. This is the University of New Hampshire, taken from a drone, relatively snowy habitat, kind of less and less each year, but good skiing up there, a beautiful place to live, and it's right in the seacoast, New Hampshire, right there, our 19 miles of coastline, which we prize. But of course, Maine is right there too, so you can access the ocean pretty easily. It's a nice place to live. And then I did spend a number of years in South Africa, and I'm still actually on the faculty at the University of Pretoria, so I like this slide in there just to remember my roots, I guess. Here's my lab as it is / was. Carrie Fuhrer is now a professor at Virginia Tech. Here's a number of people that I'll talk a little bit more about their projects at length, so I don't even know their names, and here's some colleagues in South Africa that I like to remember. So a few different groups working around the world there. And so we really approach these questions of forest health and forest sustainability from a range of approaches using both molecules all the way up to whole forest. So kind of everything in between. So machine learning and image analysis, simulation and other kinds of modeling, lots of looking through microscopes, lots of molecular work. And so students get a range of experiences too in my lab. So, okay, so just a brief overview of some of the current research projects that my lab is working on. Really the one I'm going to speak about at length, like I said, is landscape genomics and population and community ecology in the beach bark disease system. So I'm going to leave that one for now. We've also thought quite a bit about top-down, bottom-up, and chemical control in the emerald ash borer system. So here's a picture of the emerald ash borer, and it's one of its parasitoids, Tetrastichus plantagenesi, which some people might have heard about earlier today. And this is Todd Johnson, who's now an assistant professor at Louisiana State University, who looked at responses and, well, constitutive and induced defenses in ash across a range of tree sizes, so in both white and green ash, to ask the question of whether the chemical defense metabolome differs, and whether susceptibility to emerald ash borer and then subsequent susceptibility of emerald ash borer to parasitoids differs across those different sizes, classes of tree, and whether that can be linked back to tree chemistry. That's kind of an ongoing project. Some preliminary data suggests that constitutive defenses defenses, we'll call them, differ across tree size in some pretty dramatic ways here, but we're working on the HPLC analysis at the moment, so I can't really present that now. This is Liz Clifton. She's a postdoc in my lab working with the USDA APHIS group out of Massachusetts, thinking about phenological mismatching between the emerald ash borer and its introduced parasitoids across that climate gradient. So you can see she actually works in the Midwest, so from Minnesota down to Missouri, trying to understand why we see differential success and both in establishment and efficacy of some of the parasitoids that have been introduced to control the emerald ash borer, asking whether phenological mismatching is part of that story. Bree Aflag, who I mentioned to some people is now a high school chemistry teacher, so very much trying to get her through her PhD. She's just about done, but ABD, but she has done a number of projects. She actually did a master's with me first, examining parasitoid searching behavior and establishment success in what we call simulated aftermath forests. She did a bunch of treatments in the field, trying to understand how parasitoids find their host insect, in this case, emerald ash borer. And then she got really interested in the potential synergies and potential incompatibilities between chemical and biological control. So part of that was understanding how emamectin benzoate, which is the sort of systemic insecticide you can inject into the tree, translocates within trees, both in healthy trees and then trees that have already begun to decline as a consequence of emerald ash borer galleries, which turns out to affect the way that chemical is moved in a tree pretty dramatically. It creates little pockets of really high concentrations of chemical and some pockets of very low concentration, which influences where emerald ash borer can colonize, and then how that affects parasitic communities downstream, and also looking at some non-target effects. So yeah, this is Caroline Tanaski. I know the names don't mean anything to you, but she studies the southern pine beetle, and we call it the not so southern pine beetle because when was it? 2012, I think. It jumped from its northern range edge in New Jersey pine barrens to the pine barrens of Long Island, about a 200-kilometer jump. And now, as a course of some first sampling, we found it as far north as Ospey, New Hampshire, which is several hundred kilometers north of that. It's well established in Albany, New York. It's well established in Cape Cod, and actually starting to kill trees in both of those places. So it's not the southern pine beetle anymore, and this is really a consequence of changes in extreme winter temperatures. So in years where it doesn't get cold enough to kill enough of the population will persist, and if that happens too many years in a row, it can actually get built up and potentially outbreaks. So she was interested in how the associated communities of insects and a little bit of fungi, but mostly insects, differed in the Long Island populations relative to historical data set we had access to in the South. And it turns out there are some interesting differences, but actually there's quite a few similarities as well. So we'll talk at great length about that now, but we study southern pine beetle. A little foray into a subnivean community study, looking at using pitfall traps underneath snow and in the summer to compare communities of active arthropods underneath snow versus the summer sampling period, and to basically try to understand whether foraging in the winter was an important part of the food web dynamic in northern forest systems, because there is quite a lot of activity in that very small area of space that forms between the ground layer and the snow. And if that is important, then the loss of snow pattern duration and stability could be really important to changing food webs. And anyway, he was very good at getting some press for his work too. So he got a Science Friday piece, which I had actually nothing to do with, so kudos to him. But a pretty cool project there. We're also looking at using the EDRR, the Early Detection Rapid Response program of the U.S. Forest Service. So the Forest Service deploys these lingering funnel traps all across the Northeast anyway, and I think even broader than that, at areas of high risk, like ports, where people are moving things around to try to catch insects that may be just establishing or about to establish, potentially establishing in our forests prior to them becoming a big problem. And we realized, of course, that these cups contain a lot of pathogens that are either brought by the insects or just floating on the air down into the trap liquid. And we decided, well, maybe we could sequence that trap liquid and get a better understanding of fungal pathogen pressure and biogeography. And so we've been working on that. That turns out it's pretty trackable, pretty easy to do. It requires a little bit of finagling, but it can be done. And so really, I'm going to now jump kind of into the system that I've spent more time than I guess any other studying. This was the subject of my own PhD at Dartmouth College. And then I left it for a while I was in South Africa, but I couldn't leave it for too long. Came back to it once I got back to the Northeast, but it really is an important disease in our part of the world. I know that beach bark disease is not part of Georgia's landscape, which you're probably happy for, but you do have some nice, big, beautiful beaches, I understand it now. So I'll just give you a little bit of a background on this system. So beach bark disease is we call it a disease complex. So we call it complex because it involves multiple players in different permutations, basically. So what we call the initiating agent of the disease is this invasive scale insect. So it's a really tiny thing that's about a half a millimeter to a millimeter in length. That's an individual female scale insect living in this. She produces this kind of copious wax around her to protect her from the weather and from predators. But underneath that, if you tease it apart, there are thousands and thousands of these female insects that are all parthenogenetic, so there's no males known for the species. They're legless at adulthood. They have this crawler stage that had legs when they're first instar and can kind of crawl out on the tree and float to another tree. But this was introduced into Nova Scotia in around 1890 on imported European beach stock, common story. And it's believed to facilitate the initial attack by one of two fungal pathogens, which I'll talk about now, but we don't really understand the nature and full character mechanism of that interaction. So it's still a bit of a mystery, even 134 years later, but we're working on it, again, I think. So the two fungal pathogens are both in the genus Neonectria. There's Neonectria vaginata and Neonectria ditissima. And they actually look identical. You can't tell them apart. They produce these perithecia on the trees, these red little globules. They're quite tiny, but they're actually pretty beautiful if you like that sort of thing. But bright red on the side of a tree, and sometimes quite abundant. But anyway, you can't really tell them apart in the field. You can measure spore length, and if you measure enough, then you can detect slight differences in the spore size between the two species. But ecologically, they differ in some really important ways. So, for example, Neonectria vaginata is only known from the beach bark disease complex. We don't know where what its native host was. It appears to be a native species, and it spread. It was sort of for a long time be presumed to have been in conjunction with the beach scale. So this was originally thought of as an invasive species, but it doesn't seem that that's true. But it's a host-specific beach bark disease object. Neonectria ditissima, it has an extremely broad host range, so it's dozens of species and families of trees, not dozens of families, but many families, dozens of species. It's widely distributed really all across the world, and partly because it's a pathogen of apple. So it's even present in like New Zealand and South America as a pest of apple, but also on lots of forest trees all through Europe and North America. So beach bark disease has been relatively slow spreading, so color coded by how long the county has been infested. So you see the longest infected counties are in Nova Scotia, or I guess provinces in that case. And then there's an active expansion zone in Wisconsin and parts of Canada and Michigan, and then West Virginia, more recently infested. And we can use this as a temporal gradient in infection duration, which is obviously confounded with climate if we go this way, but you can control for climate if you think about across latitudinal lines, or at least climate zone lines. So we've used that sort of approach to try to tease apart the effects of duration of infestation from the climate gradient that otherwise is confounded with this spread of this disease. So one of the important components of beach bark disease is that, unlike chestnut blight, beech has not really gone anywhere in the context of this disease. So it's a relatively, dramatic initial effect of the disease. Lots of trees die, but beech has largely maintained its basal area, even in long infested forests. And so you might ask why that is. So this actually does come from my PhD. So I'm still showing this graph years later. That might happen to you if you're in your cards, right? But this is using forest inventory analysis data and basically looking across that same duration of infestation from 0 to 70 years at that time, calculating an estimate of baseline relative mortality. So how steep is the mortality curve effectively from one size class to the next, if that makes any sense. But anyway, you can see that the mortality rate, relative mortality rate, goes from about 18% on average in the uninfested forest, way up to almost 30%, so almost doubling in the infested forest, whereas sugar maple, an ecological corollary in some ways, doesn't really change that much across that same gradient. But if you look at estimated equilibrium sapling abundance, that also increases dramatically. So this is what we call compensatory improvement. So beech has a really strong propensity to produce root sprouts, and also is a masting species, so produces lots and lots of beech nuts every other year, as it turns out. But they're really shade tolerant and really hardy. The deer don't like them very much, and so beech regeneration does really, really well. And it basically replaces the lost biomass from the adult beech trees. So, but the consequence of that is that we have a sustained shift from a big sort of less dense forest to a smaller, denser forest, right? And so we still have beech, but the size structure is completely changed. It's now a small, dense forest, almost not really a thicket. It does grow through the thicket stage, but then it becomes, what you would recognize as a closed canopy forest, but it's a much smaller forest than it was before. And that has really important ecological impact. So, for example, adult beech trees produce copious beech nuts. Younger beech trees less so, although they do produce lots of nuts. Here's a thicket. If you look at biodiversity in the arthropod fauna underneath the thicket, it's much reduced. You can imagine foresters hate it because how do you grow a more valuable species under a thicket like this? It's actually really hard to get rid of. You get this beech snap, and then bear reproduction is really closely linked to mass production. So we care a lot about the production of this late season fall protein rich resource, and we don't want to lose it, or at least I don't want to lose it. But we have to recognize what the ecological impacts of beech bark things have been. I think I'm maybe mostly skip this, but we have a sort of side project trying to apply that same logic from the FIA to a bunch of different pests and pathogens across the range. Well, this is mostly focused on the Northeast, but here you can see the list of our forest threats. So chestnut blight, so note death, and others, spongy moth, trying to use that same equilibrium conception of forest dynamics to understand how things like biomass and carbon sequestration capacity change with changing demographics of tree species as impacted by these forest pests and pathogens. I'm not really ready to share this because we don't have the results yet anyway, but that's one, I guess I threw the slides in at the last minute, maybe I should. So we really want to understand some of the finer scale interactions of each part of the system. And these are the 4 questions that I'm going to present to you. So one is, how does fungal species identity, whether it's Neonectria vaginata or Dentissima, drive disease severity at the landscape scale? And then how do bark endophytic communities play a protective or facilitative role in beech bark disease expression? So there's lots of other fungi in those in beech phloem. Does that protect or enhance the effects of beech bark disease? We use population genomics to ask the question about colonization of trees by the Neonectria vaginata. And then I'll talk a little bit about bark-mediated defects. So that's just a little bit of an outline I'll talk about here. So I think I might have even made this slide during my PhD, and so it's this an original conception of beech bark disease. Here's the host, Magus grandifolia. Here's scale insect, a little orange insects under the white wax, the invasive species component of this, and then one of 2 of the Neonectria pathogens, and end result is this kind of cankering disease called beech bark disease. But then I realized, well, it's actually much more complicated than that. We need to think about it in a pairwise way and a holistic way, because all of these things are interacting with each other in some pretty important ways that we don't fully understand. And then, of course, so the environment, small matter of the environment, small matter of other microbes that are present in the system. And so we decided, okay, well, just briefly, this was sort of the original conception of the change in the abundance of scale insects and the fungi over time since initial infestation. So remember, there's this gradient, and we call the initial infestation the killing front, or so there's the establishment forest, then there's the killing front, then there's the aftermath forest. So in the killing front, you get really rapid increase in the scale insect density, and about a decade later, you get an increase in the fungal pathogen load, which tends to be dominated by Neonectria Dentissima, the native host generalist. But then Neonectria Dentissima crashes and almost disappears from the system according to the literature. And Neonectria vaginata comes in and sort of dominates from there on. And that's been the paradigm for like 50 years. But we wanted to understand if that really was the case and use some more, I guess, modern tools. And so we used ITS for amplicon sequencing, so metabarcoding basically of some hundreds of, about 150, I think, beach flown samples from across this range. And this is Eric Morrison who did a lot of this work. But if you look at the distribution of the two species, there are some, Neonectar vaginata is somewhat dominant, but there's lots of Neonectar datissima in orange, and then there's lots of places where both species are dependent. So really the only place we found only vaginata was in the mountains of North Carolina. Every other place had either both, well, both species present. And so Neonectar vaginata is it's easy to mistake it for the dominant species, because if you sample spores from the bowl, they're almost always vaginata. But it turns out that's the consequence of just it being much more prolific spore producer. And datissima is present in the bark at pretty high rates. And so if you look across tree decline class, and this is just scale, 0 is uninfested, 2 to 3 being looking terrible. There is a pattern here as well. So Neonectar vaginata is sort of present across all of these. These are all infected trees, just the decline class, it still looks good. You find, almost 80%, but doesn't change that much across this decline class. But Neonectar datissima almost always co-occurs with vaginata, sometimes even in the same little bark plug. And then as the tree declines more and more, it becomes more and more abundant. So it seems to be somewhat specialized on more heavily declining trees, but that's what we found. So, but 42% of the trees had both pathogens. So that's already interesting to me. And there's some evidence of environmental and climatic structuring. So we used joint species distribution modeling and through a bunch of different environmental variables added to ask whether Neonectar datissima incidence and severity and the two pathogens were correlated with any of these. This is a correlational analysis effectively. And it turns out that growing degree days predict stronger presence of Neonectar vaginata, so it tends to be a little bit of a warmer adapted species. Datissima is a little bit more cool adapted. Vaginata does respond positively to duration of infestation. There's slight positives for each pathogen with tree size, and they're both interestingly negatively correlated with beach scale abundance, which is kind of surprising considering that that's thought of as the facilitator for this whole system. So we decided to go into that a little bit more. And we'll know this work. This is Cameron Stouter out of Matt Cassin's lab, and my lab sort of worked together on a project looking at mating type. Trying to ask why vaginata was more prolific spore producer. I guess the upshot of that is that both of these fungi are capable of are heterozygous. Let's get that better. It doesn't seem to be described why. It doesn't explain why vaginata produces more spores. So we already had all this bark material, and we've already done metabarcoding. So we decided to look at the other things that occurred. So we used indicator species analysis and discovered about 800 ASVs. So that's amplicon sequence variants. So not species, but different signals of different DNA at the ITS4 region, which discriminates among fungi pretty well, depending on the group. And we found a bunch of stuff, but the three I'll talk about are two microbes that are known to be present in the system from the historical literature. So this one, C. rosea, is an entomopathogen. Really didn't find very much. It showed up in a few of our samples. There's also a fungal parasite in the system. It's a fungus itself, but it feeds on Neonectaria. That also is very poorly represented in our samples. But Fusarium bovida is a species that was really occurring across the range in quite a lot of our samples. And that's in the literature anyway, a putative entomopathogen. So we wonder if that's part of the negative feedbacks with scale insect density is the presence of this fungus that could be feeding on the scale insects, for example. So it's an interesting thing to think about for future work, not definitive in and of itself. Okay, so I mentioned that the historical paradigm really is that there was a single colonization event of Neonectar vaginata up here and really over here in Nova Scotia, where where the vaginata learned to learn, in an evolutionary sense, learned to exploit the interaction between beach scale and the beach host, and then it kind of spread with the spread of the insect. Really don't see Neonectar vaginata outside of this range, but another possibility is that there have been multiple colonization events throughout the range. That's also these are two alternative hypotheses. Is it a pre-adapted trait that keeps happening wherever the scale insect finds itself, or was it a single event and subsequent spread? So for this, we used population genomics approach. So this is based on SNP analysis of 74 genomes. And we found pretty weak evidence of spatial structure. There is a little bit of population structure here if you really squint, but you can see it kind of looks like a big mass of points. But what we really did find in pretty strongly was this what we call isolation by distance. So the similarity of genotypes decline as a function of space between the samples, distance between the samples, pairwise geographic distance. And to me, that is really consistent with this hypothesis. So a single colonization event that subsequently spread with the scale insect as opposed to multiple events, which would probably reflect population structure that we would expect to be a part of the system if we were living on a native host somewhere. Okay, so that, so supports that hypothesis. All right, we did some GWAS. Let's skip that. This is still work in progress. So maybe from the beach bark disease system, the last thing I'll talk about is this idea of bark-mediated negative feedbacks. And I'll explain what I mean by that. So we already saw in the correlational analysis that scale insects and Neonectaria actually are negatively correlated at the stand scale, which again is a little bit surprising based on our historical understanding of the system. There's Ken Winstein and Santiago Rivera-Reyes working on this question. And the other thing that we see in our forests, and you might know beech is this really smooth bark tree, and we do too. There is some resistance in our forests, so there are some nice, smooth, big, healthy looking beech trees. But there's also a range of bark phenotypes. So there's cracking and checking, and there's cankering, and there's deep cankering, and a lot of this appears to be a consequence of past disease in its different conformations. So this is definitely a deep cankering response that's the trees kind of survived and grown over this too, but some of this fissuring also seems to be a response to earlier infection one way or another. So we wanted to understand that a little. So one thing, we started with microscopy. It turns out Ken was really good with microscopes and in using a microtome to thin section. This is not his picture, that's the awesome picture if it was, but he also used some micro CT scanning, trying to look at the structure of the bark itself. This is a scale insect, if you can believe it. In rare instances, we were able to actually trace the stylet as it penetrated the phloem into a single paraderm cell, but it turned out that was really hard to do. So we chucked it for the subject of a master's degree, which is probably the smart thing to do. But these are stone cells, and it looks to us like the density of those stone cells influences or correlates with the resistance to the pathogen. So that's pretty interesting. But and then if you look at some of this impacted bark, you can see like it's just totally disorganized and messy. Here's what we call a microgall at the tip of the stylet of the insect that's pretty common and described in literature. And so really an entirely different reorganization of the tissue. So a tree like this, from the perspective of an insect or a pathogen, looks entirely different from that smooth bark, nice tree with beautiful flow. And so we did some inoculations. I'll jump through the methods here, you know that, but so we infested rough bark trees and smooth bark trees, either small or large trees with scale insects, and measured their growth on the tree, using eggs, so we collect thousands of eggs, it turns out, that's all eggs. Collected from trees in our area, and then can develop some image analysis algorithms to do this without having to look at every picture, pictures of everything. We did inoculations with a number of, well, both different pathogens and actually different strains across different scale insect densities. And I'm gonna give you the results as opposed to and we measured lesion density, or sorry, lesion size, but the one important piece is control trees just inoculated with agar didn't really produce much of a lesion. In trees that were infected with neonectrias had a necrotic zone, an area of active infestation or infection, and then what we're calling host response zone, which we feel a little bit confident about because it's bordered by the suberized layer that seems to suggest that the tree is sort of protecting itself at the border of that zone right there. So that just becomes, and then we did some methyljasminate inoculation or painting with methyljasminate and salicylic acid to try to induce tree defenses to see if that was part of the story. So I'm going to give you the results very quickly, and this is 3 master's degrees in 1 slide. So this is just the boiled down version. So this rough bark, which is a consequence of past disease, is actually terrible habitat for both the scale insect and the fungus. So that's a really important negative feedback in this system, from my perspective, and I think tends to be, and limits the spread of the disease locally. When we co-inoculated with both neonectar pathogens, we got lesions of intermediate size. There's really no evidence for synergistic or antagonistic effects between the 2 pathogens. So the fact that they co-occur is interesting, but not necessarily earth-shattering in terms of the dynamics of pathogen infection in the system. Neonectria lesions were smaller when in close proximity to established scale insect colonies. So again, this is supporting the idea of a negative feedback between scale insects and fungi, which I think is one of the reasons that we still have beech in our forests. Negative feedback, limiting the spread of the disease is really important. And then vaginital lesions were larger than Ditissima, but in Ditissima, the generalist elicits stronger host response. And so probably those things are related, and maybe the stronger host response is because Neonectria Ditissima has been present in the system for thousands of years, and there's been opportunity for selection. And then just briefly, both methyljasminate and salicylic acid suppress scale insect establishment, but Neonectria grew better in the presence of methyljasminate. So tree defenses matter, and we don't fully understand that yet, but we will at some stage. So our most recent, now Santi has transitioned to a PhD and wants to understand transcriptomic approaches to understanding beech bark disease interactions, sort of on the front end of that. So we'll talk more about that, but it's excited to get going. And I don't want to take too much time. I think I'm pretty close to the end of my time here, but I'll just briefly introduce you to this nematode, mysterious disease killing American beech or stressing American beech, let's say, not necessarily killing at this moment, but it turns out it's a nematode. As far as I understand, you don't have it yet in Georgia. I hope you don't get it. You don't want it. It really is. I mean, it's spreading incredibly quickly, and it produces this banding in between the leaf veins, the lateral veins of the beech leaves. So it's a foliar disease, a foliar nematode, which is very strange because I don't know of any other foliar nematodes. The only one, there's pine wilt nematode, but that's not a foliar issue either. And so this Litholinchus carnatii, subspecies McCannii, although some people are doubting the subspecies designation, they think it's just more or less the same as a Japanese nematode. So it's thought to be introduced from Japan. It overwinters in the leaf, in the buds, the bud scales, and the density of these things is absolutely incredible. But abortion is one of the big problems. The trees will just not foliate at all in the summer because the buds are basically killed by this nematode over the winter. So we're actively reorganization of the tissues. This is not my work. It's Oliveira, the ARS, So this is just the time series of the spread, so it was originally detected in Ohio in 2012. There's 2000, we're already to 21, 22, 23, and now it's in, I think it's 13 states and part of Canada, but it's moving very quickly and it's moving this way, which coupled with other evidence to me suggests wind vector. It's moving too quickly to be animal vector or insect vector. And people were originally saying don't move beach material or wash your boots, but I mean, it's just moving so quickly that it's got to be wind. So, and of course, this has potential to interact with beach bark diseases in pretty important ways. So we're working on that, largely using dendrochronology at this point. So trying to understand how growth rings change as a function of infestation, both severity and duration with the nematode in beach bark disease and infested and uninfested trees and stands. So that work is kind of ongoing and I don't have results yet. So I'll end there just by saying, you know, our forests are still maintain a high level of resiliency. So I have faith that the forest will persist, but you know, as these things start to overlap and interact in ways that are difficult to predict, we really keep an eye on this. So any questions? Happy to take them.